Thank you, Leanna, and thank you and your department for the invitation to come and share with you. I recognize everybody's coming back from the holidays, so I hope we can keep it light and you feel like this is a nice introduction back into your work week after a long weekend. So before I begin, I just wanted to thank my team who contribute to all of the work that I'm going to tell you about today. The different versions of the team, and certainly some of the alums have contributed to a few of the stories that I'm going to tell you. So I just want to acknowledge them for all of their hard work. They're a really great group of people to work with, as well as the funding that makes all of this work possible. So I have a few goals for our time this morning, and I hope I can get through all of them. I think at least the first four we should be able to cover. One is I want to give you some context to some of the disparities that exist in Alzheimer's disease that my group has been interested in, and convince you that proteomics is an important problem or an important tool that can be used to solve this problem. I want to share with you how proteomics measurements are made and give you an idea of how we make sure that our measurements are really robust. And then I want to give two different case studies. One, I want to show you how we've tested the notion that there are molecular contributions that may contribute to disparities in disease, and then share an approach that we've been using to look at pretty much like biomarker discovery types of efforts in diverse populations. And if we get to the end, then I can give an overview depending on how time goes. Okay, so let's start at the beginning. One of the things that was really interesting to me during my postdoc and as I was beginning to start my laboratory was digging into Alzheimer's disease literature and discovering from the Alzheimer's Association that there were disparities that exist with this disease. And while this disease is especially devastating for all communities, what was interesting to me was that within the African American and Black community in the US, that there are 2 to 3 times more likelihood that these individuals have Alzheimer's disease than non-Hispanic whites. For Hispanic Latinx individuals, that would be like 1.5 to 2 times. And so I was intrigued by that. And I also was, concerned by the fact that these disparities impact this community in a number of different ways. One, you can see that while African Americans are less than 14% of the population, it was estimated that a third of the total cost for the treatment of Alzheimer's and other dementias was burdened by this community. And when you look at these financial costs, these are, really expensive industries in terms of financing to care for someone who has been impacted by the disease. So oftentimes people are putting loved ones in facilities or bringing nurses into the home, and especially within this community are giving up their jobs in order to care for loved ones at home. So there's an economic disparity here as well, in addition to the prevalence. What's also interesting to me is the fact that there are a lot of reported experiences from racial and ethnic groups that have to do with discrimination in our healthcare system, and as well as related to biomedical research. So what you see here on the left of the screen is that a number of racial and ethnic groups report frequent discrimination when they're seeking healthcare. And then what you see on the right side of the screen is that a number of groups, especially within the African American Black community, believe that medical research is biased against people of color. So I was also interested in why this was occurring, because what I recognized when I started to look at basic science research studies is that there was a lack of diversity within that context. And so there have been a number of groups, and especially within the NIA, that have spoken to the need for diversity in Alzheimer's disease research across basic science to clinical trials. And the NIA has been very responsive, I think, into creating opportunities for individuals to work within this space and have funding. So one might ask, what are, some of the reasons why individuals in the African American Black community might not participate in research? And one of them is what I just alluded to, which is this lack of discrimination or mistrust that comes from interactions with medical professionals, researchers, or big pharma companies. And this is not only something that has a long historic context, but it also, based on the report, is something that is continuing to happen and persist in society today. So while that's one of the biggest reasons, I think there are other reasons as well, which are illustrated here. So there could be accessibility issues, whether that is getting access to the study or having access to the care that's needed to participate in the study. There are fears that individuals have about the research or not wanting to be a guinea pig in the research and want to understand how it actually is going to benefit them, and making sure that they're being treated fairly when participating in the research, and considerations related to their cost and time. And these are not all inclusive, but just some of the reasons why one might not participate. So as an aside to the rest of the work that I'm going to tell you about, I was really interested in wanting to help and do something about some of these barriers to increase recruitment for research. And in part, I'll share, why that motivation exists in a few slides. So I became involved with this study called Recruitment Innovations to Enhance Diversity in Alzheimer's Disease Research. And it was led by Dr. Jennifer Lingler at University of Pittsburgh and some of my other colleagues here who are shown on the slide. And the idea here was whether or not we could understand what were the motivations to participate in research from individuals that were in the Alzheimer's Disease Research Center. So specifically, we were focused on African-American Black participants who had agreed to be in the studies and their caregivers. And we wanted to learn from them, despite different barriers that they may have had to overcome to be in the study, what were the motivations that encouraged them to participate in the study. So we conducted a number of interviews, and from those interviews, we captured narratives of the types of personas that persisted across the people in the center. And we were able to take those narratives and turn them into recruitment materials. So what you see here are examples of Q&A brochures that would be handed out at a doctor's office, or that we would take to community events or have on display in different areas, in churches and community centers. And they have the authentic stories of the individuals that we interviewed. In some cases, they agreed to share their actual photos. And so here you can see Annie and her grandmother in this photo. And so those are actual individuals. And then in other cases, we might have used, stock photos or actors who were representing personas that didn't want to agree to consent with their photos. And then from that, we were able to conduct a number of focus groups to see how well those materials resonated with individuals in the community. So in addition to these materials, we also had videos. And during COVID, we were able to transition these videos into a social media campaign to see how effective it was with increasing research participation at Pitt. And we learned a lot. We learned what are some of the things that are triggers for individuals that turn them off when they see words like research, or how certain imagery is displayed, or if it's too hard to read, what's the likability of the images and the way that things are put together, and whether or not the messages that were shared actually encourage them to want to participate in research. And with that, we were able to adjust the materials to make them more effective. And then we also transitioned the work to Nashville. And so part of the motivation here was Nashville is located in the south of the US, and it's closer to some of the historic events that have occurred in this community related to mistreatment from the medical system. And so we wanted to see whether or not there were any geographical differences in how people receive the different materials. So that's an ongoing work that I've been doing here at Nashville in collaboration with our Qualitative Research Core. So all that to say, one part of the problem is making sure that there is inclusiveness in the individuals that are participating in research, but also understanding that there are many factors that have to be considered when we want to think about reducing disparities in this and other communities. So here is a beautiful framework from the National Institute of Aging that gives an idea of all the different ways that we really have to think about an individual's lived experiences in order to reduce disparities. So understanding, what their experiences are as related to discrimination, criminalization, environmental toxins and exposures, what are their lifestyles, what kind of things do they eat, what kind of access do they have to healthy foods, insurance, socioeconomic status, just so many different things, genetics, that go into really understanding how all of these different factors interplay with each other, that will ultimately impact the types of things that we see at the biological level, which is a lot of what I'll be sharing as we move forward. So I entered in, with this kind of framework that we have seen before from Alzheimer's disease, which is understanding that there are a number of factors that increase risk for Alzheimer's disease pathogenesis. So regardless of who you are, if you have other comorbid conditions like high cholesterol, hypertension, dyslipidemia, these things all can increase your risk of disease and contribute directly to pathological hallmarks, as well as different genetic factors. And so while we're all familiar with ApoE, I also wanted to highlight that there are specific genetic variants for genes like APCA7, which is the ATP cassette transporting protein that transports lipids in and out the cell. And so some of these variants have been demonstrated to have potentially an increased risk or increased effect size for the African American community compared to ApoE, which has had mixed results. And so understanding these lived experiences, comorbidities, and genetic factors is really critical to how we paint the picture of what Alzheimer's disease looks like in diverse groups. And so you all are familiar with the pathology that happens in Alzheimer's disease. And so I'll just share that one of the things that was interesting to me were a couple of studies that started to look at whether or not there were differences between non-Hispanic white groups and African-American Black groups. And so the first on the top is a study from Dr. Barnes's group at Rush University that was looking at pathological hallmarks and the proportion of different types of dementia in African-American and non-Hispanic white individuals at autopsy. And here what they found was that there were more cases of mixed dementia in the African-American participants relative to the non-Hispanic white participants. So there were things like infarcts, Lewy body disease that were also occurring in addition to Alzheimer's disease. And later, what they showed was that it really depended on where the participants were coming from, if they were from the community, or if they were from the clinic, in terms of whether or not these results replicated. But that was interesting to us. The other piece that was interesting come from a number of small studies that are represented here on the bottom, which look at cerebral fluid markers of total tau. And so here what you can see is across the age spectrum, the levels of total tau are lower in African-American adults compared to non-Hispanic whites. So this to me was really critical because if this is in fact reproduced at large scale, what it suggests is that a number of individuals will go undiagnosed or underdiagnosed for Alzheimer's disease if the biomarker levels are set based on where they reside for the non-Hispanic white population. So I think these were all reasons that showcased to me that it was important to think about how to use the expertise that we had in my lab, which is proteomics, to begin to understand Alzheimer's disease in this population. But in order to do that, I first started to see, well, what had kind of been done before with proteomics in diverse communities. And so these were some of the major studies that had been out several years ago. And what you notice is that the participation of non-white participants is pretty low in a number of these studies. So that was one challenge. And even at our own center, when I was at the University of Pittsburgh, it also was a challenge. So in our center, we had about 1,200 participants. We had longitudinal plasma samples banked from 1,200 participants. But when we looked at what was available from African-American participants, we only had 130 plasma tissues. And we looked at, when we looked at postmortem brain tissues, there were only about 6 to 10 cases or controls that were coming from this community. And so I was faced with, how do I move this research forward if I don't have a really statistical good size of samples in order to work with, while also wanting to minimize introducing batch effects by trying to combine different brains from different centers. So we decided that we would just start with what was available at Pitt, generate some pilot data, and then try to go from there and see if we could get our hands on larger numbers of tissues. So that's kind of the context for the work that I'm going to tell you about. So before I tell you about what we did and what we learned, I want to just teach you a little bit about how we actually make proteomics measurements and what I mean by proteomics in case you are not familiar. So when we're talking about proteomics, we really are talking about the measurement of all the proteins or as many proteins as we can access from different tissues. That could be plasma tissue, cerebrospinal fluid, postmortem brain tissue. It could be cells. It could be, whole organisms if you're especially working with animal models. We extract all the proteins out of the tissue, and then we actually make our mixtures more complex by using chemical enzymes to take the proteins and digest them into peptides. And so we do this because the peptides are easier to separate and to analyze by mass spectrometer instruments. We can do different types of chemical strategies in order to make our measurements more throughput. And so I'll share with you what that looks like. But essentially, we take these peptides through this cycle of separating them, introducing them into the mass spec, and then we use database searching tools and algorithms, since the genome has been sequenced, in order to match our experimental data with theoretical data of what these peptides and proteins hence are that are in our samples. So from the mass spectrometer, we get some simple readouts, although these are actually complex measurements. And so the simple idea is that we take our sample, it gets introduced into an LC, and peptides come off of the LC and go into the mass spectrometer. And the first readout that we get is a mass discharge value of each intact peptide ion. This is in the gas phase, so all the solution has been removed. So we have that information. We then take that peptide in the gas phase and we subject it into high energy collisions, and we break it apart. And so we measure the masses of those fragments. And so that's the bigger spectra that you see here. And we piece it back together like a puzzle. So we can figure out the amino acid sequence of the peptide that was broken apart. And then from the database information with the first readout and the second readout, we have an idea of the sequence. We know if we've modified it or not, and we also can identify which protein it came from. The third readout comes from chemical tools or strategies that we also use in our workflow, which give us information about how each peptide changes relative to from one sample relative to another. So in this example, and the ones that I show, we were interested in differences between cognitively normal individuals, those that have Alzheimer's disease, and from the different racial ethnic groups that we were studying. So the way that we make these measurements high throughput is in a couple ways. One is we introduce chemical tags to the peptides. So peptides have primary amines that are present on them. And so we react them with a tag like this that's commercially available called a tandem mass tag. And so it has a reaction site, which is the N-hydroxy succinimide ester that reacts with the primary amines. And then what you see is this mass balance and reporter ion region. And so these regions are designed strategically so that the manufacturer produces have 6 different versions of the tag that have heavy isotopes that are embedded in different portions of the mass balance and reporter ion region. And so once this tag is reacted with the peptides, what happens is they all have the exact same mass. So when you introduce them into the mass spectrometer, you get one peptide peak, but that peptide peak contains 6 different samples. And then after you fragment this peptide, you cleave this mass bond between the mass balance and reporter ion region, and you release this reporter ion that differs in increments of 1 unit. And so here's where we get our quantitative information. So when I first started my lab, the state of the art was like you could do 2 samples. And then over time, and now we can do 18 samples that we can mix together in a single experiment. So that's one way that we make our measurements high throughput. The second thing that we do is we incorporate automation. So I have a team of individuals who help to bring in this Biomex system into the lab, which is the automated robotic liquid handler. And so here what we do is we have our samples on a 96 well plate, and then this robotic arm goes through and it picks up and dispenses liquid across plates to do our chemical reactions. So that whole cycle that I showed you, sample prep for proteins to get to peptides, to get to the tags, and to get mass spec ready, we're able to incorporate on this biomic device. We also have a second arm that can do larger volumes. So this is an 8-pod that can pick up to 5 milliliters of volume. So this is a pretty fancy robotic liquid handler, but the idea is that once we actually get to having large numbers of samples, we can be able to handle and prepare close to 1,200 samples in a single run by using the multiple plates that are available on this particular system. So we have ways that we make our measurements high throughput. The other thing that we do is we try to make sure that the data that we collect has many aspects of quality control incorporated into it, so that as we're acquiring data over months at a time, we can make sure that the data is all of good quality. So we think about that from the sample preparation point of view, where whether we're working with plasma or brain tissue, everywhere in the sample prep process, we have quality control samples that we've embedded and we know how they should behave. And we're monitoring that as we're preparing samples. So we go from one step to the end and then go from there. Thank you, Leanne. Do you need to remove someone from the meeting?
Yeah, I think we're being like, I think random people are getting in the call somehow.
Okay.
And I don't know how it's happening. I'm sorry.
Okay, all good. I'll work around it. We're good. Okay, so we have the sample prep piece that we look for quality controls. And then the other thing that we do is once we introduce the samples into the mass spec, we make sure that the mass spec is performing optimally. So we have about 8 different measures that a postdoc in the lab, Dr. Kyrie Patterson, came up with. And these 8 different measures allow us to monitor how peptides are performing overall. And there's certain internal peptides that we look for to make sure that they're meeting our specs. So anytime we have a value that is out of specification, like down here, then we know that we actually need to stop our runs and adjust something with the instrument so we can keep going. Okay, so we pretty much have a process where we assess quality control as we're acquiring samples throughout the day. And in real time, we can see whether or not we should keep moving forward and collecting data on our actual samples, or if we should stop and fix something. And we actually know where we should fix something down to the different parts of our process. Okay, so I hope you feel like you understand a little bit about how we make proteomics measurements. So now I'm going to show and share 2 different case examples of how we use these measurements to evaluate disparities in disease. So the first was work that was led by Dr. Caitlin Stepler, who was able to take different brain regions from the tissues that were accessible from the Pitt Alzheimer Center. So for this pilot study, we were able to get access to hippocampal tissue, inferior parietal lobeal tissue, and globus pallidus tissue. And we knew that hippocampus and IPL were pathological tissues, and then the globus pallidus had fewer changes, and so we actually wanted to use that as a negative control. We also had cortex, but that didn't get evaluated in this study that I'm going to tell you about. And so what we did was, since we were limited by the African-American participants, was we then matched and randomly selected from the non-Hispanic white participants based on sex, age, and years of education, and then tried to make sure that the groups were balanced in that particular way. And so that's what we were able to work with for this study. And the idea here is in each of our experiments, we start with 1 brain region, but in every experiment, at this time, we had 10 samples that came from participants and 1 quality control. And then we would go back and run the next brain region and then the next brain region. So you can see that these experiments generate lots of information. So on the right, what you see from the Venn diagram is that across the different regions, we identify 1,600 to 2,000 proteins per each region. And some of the proteins that we identify are unique to the brain region that they come from. And then depending on how prominent that protein is across all of the participant samples, then we move it forward to actually look for quantitative differences across the groups. So this will be an example of what that looks like, where if you go to B and look at this volcano plot, here what you see are the levels of each protein. So each dot is a protein, and how that protein level changes from the Alzheimer's disease participants to those that were cognitively normal. And then on the y-axis, you see the significance of that particular change. So everything in red is a significant change. If it's on the right, it's higher in expression in individuals with Alzheimer's. If it's on the left, it's lower in expression in individuals with Alzheimer's. Most of the changes that we observed were present in the hippocampal tissue. So here we saw close to 500 different proteins that were different. As we moved to the IPL region, we saw less proteins that were changing, but a couple of players that you may be familiar with, so GFAP protein and alpha-2 macroglobulin. And then when we got to the globus pallidus, there were very few changes that were present in the proteome, which was what we anticipated. If you look at the data in a different way, here's a heat map that allows you to see how protein levels across each row cluster from the different participants, which are the different columns. And so we had two major clusters here. The first one just shows that most of the proteins that were different in hippocampal tissue were lower in expression in people with Alzheimer's disease. And there was a smaller group of proteins that had the reverse trend. I really like showing this heat map because I think it shows the power of like having big data and lots of big data. So essentially what you can see from the top clustering of the participants is that based on how the proteome changes, we can identify participants who may have been diagnosed as having Alzheimer's disease, like this, there's a purple one over here on the left side from a non-Hispanic white participant, they actually look more like the cognitively normal proteome than they do the individual with Alzheimer's disease. So I think it gives us another opportunity to look at diagnosis in a potentially unbiased way. In terms of the proteins that are observed, we then look to see what pathways they're involved in. And so here you can see that there's a lot of changes in proteins involved with signaling pathways, synaptogenesis, mitochondrial dysfunction, oxidative phosphorylation, which are all pathways that are known hallmarks that change in Alzheimer's disease. And so also you can see like the glycolytic pathway and glucogenesis pathways are other pathways where we had proteins that were lower in expression in people with Alzheimer's disease. And that's very consistent with what we know about lower glucose metabolism in Alzheimer's disease patients. So all that to say, we have a lot of confidence in the data that we generate from these studies. So the question that we really wanted to know at this time was whether or not the proteins that we were seeing had any differences in their expression based on the racial ethnic background of the participant. And so for that, our colleagues in VUMC, Dr. Timothy Homan and team conducted an interaction analysis that looked at the race and the diagnosis of the participant. So for example, for alpha-2 macroglobulin, what you see here is that this is a protein whose levels didn't change in the non-Hispanic white Alzheimer's disease participants compared to those that were cognitively normal. However, in the African-American participants, it was lower in the Alzheimer's disease participants. So one group, no change, the other group, a change. Similarly, if you look at alpha-synuclein, you can see that there is a change in one group, but the change is different in the other racial and ethnic group. And then there are examples like the ribonuclear protein up at the top, where the proteins behave the same way in both racial and ethnic groups. So it doesn't matter who the individuals are. These are proteins that are elevated in individuals with Alzheimer's disease. So again, this was a pilot study, but I think this was critical because it highlights the diversity in the disease, and that if you don't have a lot of diversity in the participants, you're actually missing information because you're not sampling everybody. But actually, it's important to understand how stable and what the spread in some of these protein signals looks like across groups. So we had generated that data, and then at the same time, we recognized that we wanted to spend more time. I'm an analytical chemist, so we wanted to spend more time upgrading our analytical workflow. And so this is where we started to pour a lot into the automation. We wanted to increase our proteome depth, so we wanted to be able to go from a couple thousand proteins to more proteins. And so there are a number of analytical things that we did. And through those changes, we were able to then improve how many proteins we could see. So we were able to get a larger number of brain samples from the Rush Alzheimer's disease cohort. So here we went from about 20 to 40 participant samples across regions. And here you can see we could see now close to 10,000 proteins from improvements in our analytical workflow. So it takes a lot to get here, but we're pretty proud of some of the work that we've been doing here. So we then wanted to know how relevant were some of these proteins in terms of being able to distinguish people that had Alzheimer's disease from those that didn't in diverse cohorts. So what we did was we worked with Dr. Heather Desir, who is a machine learning expert and also proteomics expert. And we pulled all the data from other proteomic studies that had African-American, non-Hispanic white, Hispanic participants in them. And so you can see what the racial breakup looks like from these pie charts. And because these were proteomic studies that were done somewhat similarly in the experimental design, we were able to combine the data. And each particular study measured anywhere from 8,500 to 10,000 proteins. So our process was to then go in and select protein features that were common across all of these data sets. And we wanted to use these data sets in the literature to train a model that could robustly identify people with Alzheimer's from those who didn't. And then our data set from Pitt was the test data set to see how well this model performed and whether or not the performance of the model was different in our non-Hispanic white and African-American participants. And so the results from that work is shown here, where if you look at the top a rock curve, what you see is that amyloid precursor protein is really good at distinguishing people that have Alzheimer's disease and looking at the brain tissue. But then on the bottom, what you see is that as the diversity of the cohort increases and there are more African-American participants, then heat shock protein beta 1 becomes another protein that has a really good distinguishing capability. So all of this to say that, again, it matters who the participants are in these studies in order to make sure that the potential, biomarkers that might be used to diagnose disease are going to be relevant and work for everyone. So that is great. And we continue to do work in postmortem brain tissue that we have access to. But again, that's postmortem brain tissue. So it can tell us a lot about disease pathogenesis and kind of what's happened already. But it doesn't directly translate into some of the signals that you might see from plasma that could be used for biomarker research. So we started to do some work in that area as well. And if you think about the current biomarkers for Alzheimer's disease that rely on cerebral spinal fluid or PET scans, these are great and they're really robust. However, these are tissues that are not always accessible or desired by African-American participants to give CSF. And then the PET scans or MRI scanning technology, all of these technologies have to be accessible in communities in order for them to be able to access those biomarkers. So a lot of groups are interested in using other tissues like blood and plasma in order to develop biomarkers, but these aren't easy tissues to work with. So plasma is a tissue that's very heterogeneous. We did a deep dive of looking at plasma proteomic studies, and essentially the more studies you start to look at, the less they converge on which proteins are different in individuals that have Alzheimer's disease. So that's what this shows here on the left. And then analytically, plasma is really hard to work with because of the range of concentrations of proteins in our plasma. So if you look at this graph, what you see is that there's a 10 to 12 order of magnitude difference between albumin, which is the most abundant protein in plasma, to interleukins or VEGF, which are, really interesting signaling molecules. And so we think when it comes to potential biomarkers, these are going to be things that are lower in concentration. So we have to have analytical tools to fish those out and be able to measure them with a lot of sensitivity. So this was some of the work that we did with a former student in the lab who works at Takeda Pharmaceutical. And the idea was to take those plasma tissues that we had access to at Pitt and to do a differential proteomic study, and then to use that data to also do a machine learning study to see whether or not we could identify potential biomarker candidates and how well they perform dependent on the racial background of the participants. So we had a training set of participant samples, and then we had a test set from the same Pitt cohort. Working with plasma, there are a few things that we do differently. And the only thing that I'll share is we use a column that has antibodies for the most abundant proteins in plasma. And so we can remove between 6 or 14 highly abundant proteins from plasma and then do the rest of our proteomics workflow. We run everything in replicate and we split the samples across different TMT batches. And so here what you see is that from batch to batch comparison, the results are really robust and they correlate really well. So we are confident we have really high quality data. And then as I shared, the dynamic range of plasma proteins is variable. So here the black trace shows you those theoretical concentrations of 60 of the most classical proteins that we know should be in plasma. And the orange and blue trace just show you where our mass spectrometer measures those levels. So it's very consistent. And in some cases, the mass spec is even more sensitive than what we anticipate from these known concentrations. Okay, so we applied the proteomics to the plasma samples. And you can see we like volcano plots. It helps us to visualize what happens. And so if you look at the one on the left, here what you immediately notice is that the sheer number of proteins that are different in people that have Alzheimer's disease is substantially less in plasma compared to that in brain. But then as you compare across these volcano plots, depending on how we've stratified the participants, you'll see that there's also differences in which proteins are driving the changes we see when the non-Hispanic white and African-American participants are grouped together. So we wanted to then take the information from these volcano plots, so the proteins that were different, which are above this dashed line, and feed them into a machine learning model. In this model, we had two different approaches. One was to just use the protein data, and the second was to use protein data along with sex, age, years of education, and ApoE status of the participants to see if that helped to improve the model's performance. And essentially, if we were using the proteomics data on everybody and fed it the proteins that came from everybody, then we had a 68% accuracy of identifying who had Alzheimer's disease or predicting who had Alzheimer's disease. If we added in the clinical variables, then we improved the accuracy to 74%. But what was interesting was what happened when we started to test the model on only the non-Hispanic white participants and only the African-American participants. And here we weren't able to get the performance above 70% for the African-American participants. And so that led us to the questions of, one, really the challenges of the sample sizes and needing to have more sample sizes to have better protein stability. But then also wondering whether or not the variables that we were using are actually the best variables to use in order to improve the model's performance. So years of education doesn't, equate to the quality of that education or one's educational experience. So a lot of work to be done there. So I want to share, so that was with proteomics data, but I want to share the power of omics data in general in a way that I think is can be somewhat unbiased, but I'm gonna contradict myself because I'm gonna tell you about all the biases in what I'm doing. So this is looking at the same plasma samples from Pitt, but now we're looking at lipids. So different molecules, different sample preparation, and even a different mass spec approach. So here we can take 25 microliters of plasma, we can extract the lipids out of the plasma, we put in an internal standard, and in these approaches, we have a very targeted way that we can measure lipids. But what's interesting here from the lipids is that we can find some really good lipid markers that can distinguish individuals with Alzheimer's disease. So here what you see, if you take a look at the two bottom traces, are examples of a phosphatidylserine whose levels are lower in people with Alzheimer's disease across both of the racial and ethnic groups. And then in D, you see an example of a different lipid whose levels are not the same when you compare across the different racial ethnic groups. So even though we were looking at different classes of molecules, we were finding similar behavior where there's some things that work for everybody and there's some things that look like they may be unique. So this was measuring across, we initially started with about 1,200 lipids, and then we focused our assays to about 329 lipids that had really robust signals. But what I want to share is with the lipidome as well, you could clearly discriminate individuals that were cognitively normal from those who had Alzheimer's disease using this partial least squares discriminant analysis. And what was interesting to us is if we stratified the participants, again, by racial and ethnic groups, then we could see that the individuals that were cognitively normal were more similar across those groups. But then there was a distinction in the individuals that had Alzheimer's disease. So don't ask me why we see those differences, because I don't think we know enough about the participants in order to explain why those differences, are occurring. But I think what it does show is that we have to measure, a lot of individuals and individuals from different backgrounds so we can actually converge on the molecules that are consistent across the different groups if we're thinking about biomarker discovery efforts. So I said I wanted to share sort of like somewhat of a contradiction in terms of thinking about how the experiments that we've done are biased in a way. And one is just understanding that race is something that is a social construct. And I don't want to present the misconception that the changes that we see are due to race, because race is something that we've created. And in fact, there's so many different other factors that are contributing to the biology that we would measure. So we know that what we eat influences our plasma levels, our genetics influence our plasma levels, what our environmental exposures are, and all of those other lived experiences that I described can impact the levels that we're seeing. And so it's important to think about how we incorporate all these different aspects of an individual into these types of studies where we have these kinds of power to measure biology really well. So I think for my field, who uses omics and other people that use omics, we really have to challenge ourselves and think carefully about the biases that we're introducing into our study design and even in our participant or patient selection. What kind of misassumptions do we have when we're thinking about race? Are we incorporating things like ancestry, really looking at heterogeneity across patients? I spoke earlier in the talk about making sure we're engaging diverse participants in research, and this includes in basic science research as well, so that we can have adequate sample sizes to make sure that the findings that we get from these types of proteomic studies can be generalizable. But we have to have a lot of people participating in order to do that. So I hope I've been able to share a bit about what we do in my research group. Certainly this isn't everything. And for the sake of time, I knew I wouldn't get to the equity part, but that's okay. But we basically develop a lot of high throughput mass spectrometry approaches that we apply to plasma, postmortem brain tissue, other tissues from diverse participant samples. And right now we're really interested in trying to understand why some of the disparities exist in the African-American community, but also how to make the research that we do equitable. So with that, I will stop and I welcome questions, comments, conversation before you have to leave at the top of the hour. So thank you.